Hi Hires, we are looking at Key Area 8, Genomic Sequencing in DNA in the Genome. So one of the biggest things that's actually changed in terms of genes since Mendel, when you could just basically say, oh look, this isn't being inherited and we're looking at a pattern of inheritance, is this idea that we can actually see the A, T, G, C, 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 G, G, A, A, whatever, that specific sequence, we actually can find it. Okay, and the technology, it's been around for a bit, and there's been a lot of speeding up of that technology, especially with the use of more automation and computers and things like that. But in the late 70s was when some of the early sequencing methods were developed. Okay, so Sanger's method was really quite, well, very clever. What they started with was template DNA. So the original thing you need for DNA replication. Now, if you go through the rest of your list, you'd say, right, okay, I need my template DNA, I need helicase, I need ligase, I need DNA polymerase, I need energy in the form of ATP, and I need an awful lot of free nucleotides. And the one thing they changed in it was the free nucleotides. So they gave loads of normal ones, but then they also put in some that had been modified so they couldn't actually join in to another one. So there was plenty of A's, but then there was also some weird A's, some A's that didn't fit, and plenty of T's, and then some weird T's that when it stuck in you couldn't go any further. And then they just tried to replicate it lots and lots and lots of time, and every time it hit one of these modified nucleotides it would have to stop. And then you end up with every single possible length of strand, stopping at every single base. So I'll show you that in a little doodle in the next slide. Okay, so here's my doodle space. So let's assume that you have a sequence that runs A, T, T, C, G, I don't know, a wee pile like that, okay? And so you start copying that sequence, and let's assume that we're actually not doing the copy, but we're getting the actual code for it. So I start sequencing and I get to A, okay, and then T, and then T, and then C, and I'm doing fine, and then I hit this one, and for just by chance I pull in one of these weird C's, because remember I've got lots of normal C's floating around, and just a few of them have got these weird modifications to them. So most of the time you're going to get a normal C, but sometimes you're going to hit one of these odd ones. So I end up with a sequence that only got this far, okay? And then let's say it starts again, because remember we're replicating this lots and lots of times, and this time it gets to here before it hits one of these weird ones. But then some of them might only have got as far as that, or even just the A. And so we get every possible length of code, just depending on which time you actually hit one of these weird ones being pulled in. And now they can take every single one of these, all these ones, and well, now we use slightly more complicated things to do it, but what I used to do was just put them on a gel, okay, and run the whole thing. And what we'd have is our shortest ones would be down here, so that would be an A, and then we'd have an AT, and then we'd have an ATT, which actually would be here because it would be a T, and you would just basically build your way back up with all of the different bands that you have, and you would read off the letters as to whether it was an A, a T, a G, or a C, and then you just write it down. Very clever system, very labour intensive to start with, and now we use something a little bit more techy. This would be a nice one showing the actual final detection of it. This time we're using a capillary gel and a laser that is detecting which fragment you've actually got, how long it is, and what's at the end of it. Is it a T, a G, a C, or an A? And you'll notice that in here we're also being quite clear, you still need to have that primer section, because otherwise you wouldn't be able to get your DNA polymerase to run. Okay, so if you do this and run it well, you can not only sequence individual genes, you can even do entire genomes, and that has been done, quite a lot of them. And several of them we have model organisms, so that would mean that you're taking things that you think are typical from a particular group, so taxonomic being one of our kind of main splits that we name things as. And if you have a model, then you're saying this is what we expect in a fish. Like you've taken something like the zebrafish and you've sequenced that entirely. So when you then look at, say, a trout, you can compare it to the zebrafish, and you can then have a conversation about how different it is, how similar it is, things like that. we obviously are going to do sequencing of anything that causes us real harm. So disease-causing organisms, we have an awful lot of information on them. And in the same way that if we're talking about things that do harm, in terms of crop production and animal welfare, pests are an issue that we want to try and deal with as well. Now, all of that is fabulous and it is amazing, but the problem now is that we have so much information that we need to start using technology to deal with that information. And this is the big word, and this is what it means. If you use computers to analyse the information you have from sequencing data, then that allows you to do comparisons and allows you to really see what's going on. And what we're trying to do is to find out where there's similarities and where there's differences. So bioinformatics, that's your big word. And that is the key area.